Welcome to this tutorial on the fundamental concepts related to enzymes brought to you by BIOCHEMISTRY Power Capsule series. This video pertains to the following CBME curriculum topic: explain fundamental concepts of enzyme, isenzyme, alloenzyme, coenzyme and cofactors. Enumerate the main classes of enzymes according to IUPAC nomenclature. A highly remarkable and specialized function performed by a class of proteins in our body is that they act as biological catalysts. These catalytic proteins are called enzymes, and they speed up the rate of innumerable biochemical reactions in the body without themselves getting consumed in the process. Almost all the body reactions are mediated to some extent by enzymes. Biochemical reactions and transformations need to occur rapidly if life has to be sustained. Such reactions would not have occurred rapidly enough to sustain life if this class of molecules, known as enzymes, were not present. Reactions catalyzed by enzymes would also have been possible without the presence of enzymes, but then they would have occurred so slowly that life would have been practically non-sustainable and would not have evolved even. A reaction that takes place in milliseconds would take hours to occur in the absence of its enzyme, or may not even occur at all at normal temperature and pressure. Even a simple reaction pathway would be incredibly slow or impossible. Therefore, enzymes are essential for life. The question that needs to be asked at this stage is, why do we need to study about enzymes? Studying enzymes is crucial for understanding the fundamental processes of life. By comprehending enzyme structure and function, we can gain insights into disease mechanisms, medical diagnostics and drug development. The catalytic efficiency of enzymes is extraordinarily high, and most enzymes can enhance the reaction rates by a factor of 10 to the power 5 to 10 to the power 17. A single enzyme molecule is capable of transforming thousands to millions of substrate molecules into the corresponding product molecules each second, depending on the type of enzyme. Some type of catalytic RNA, termed ribozymes, can also serve as enzymes. Most of them play specialized roles in nucleic acid molecular mechanisms, but are generally not involved in metabolic reactions. Where are enzymes located? The location and place of activity of an enzyme depends on its purpose. Such locations or place of activity may include cytoplasm, cell organelle, bound to membrane, in extracellular space, in interstitial space, vascular space and in external compartment. Most enzymes require non-protein chemical components for performing their catalytic activity. These components are termed cofactors. The complete active enzyme consisting of the protein portion and the cofactor is called a holoenzyme. The protein portion is termed a poenzyme. The apoenzyme alone is inactive. Cofactors may be either inorganic metal ions or organic molecules. Conventionally, a cofactor usually refers to the metal ions, while the term coenzyme is specifically reserved for organic cofactors. Hence, a holoenzyme is also a poenzyme plus coenzyme. The noteworthy metal ions that act as cofactors include calcium, copper, iron, potassium, magnesium, manganese, nickel and zinc. Enzymes that utilize metals as cofactors are termed metalloenzymes. Coenzymes are derived from the vitamins of vitamin B complex group. Detailed discussion on activities of enzymes that require cofactors and coenzymes will be presented in subsequent tutorials. Coenzymes can function as either co-substrates or prosthetic groups as co-substrate. Coenzymes associate with the active site of the enzyme only transiently and reversibly for the purpose of the catalytic reaction. It is changed chemically during the reaction, and after completion of the reaction, the chemically modified co-substrate dissociates away and is free to participate in other enzymatic reaction. As prosthetic group, coenzyme is bound permanently to the active site of its enzyme, either covalently or non-covalently. Dissociation of the prosthetic group results in an irreversible loss of catalytic activity of the enzyme because a prosthetic group is permanently associated. Conventionally, enzymes are identified by their trivial names, such as hexokinase, amylase, sucrase, pyruvate kinase, and so on. The suffix ase denotes the substance as an enzyme. However, a uniform system for enzyme nomenclature and classification, as proposed by the International Union of Biochemistry and Molecular Biology, is followed nowadays. In this system, enzymes are grouped into 6 major classes. This mnemonic will help you remember the names of various categories: oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. Oxidoreductases are involved in the oxidation-reduction reactions, which include electron transfers, hydrogen transfers, and reactions in which oxygen participates. Notable examples include dehydrogenases, such as lactate dehydrogenase and pyruvate dehydrogenase. Transferoses catalyze the transfer of functional group either from a donor molecule to an acceptor, or within a single molecule. Notable examples include kinases, such as glucokinase, hexokinase. Hydrolyses make use of water to cleave a single molecule into 2 molecules. Notable examples are digestive enzymes, lysosomal enzymes. Lyase split the substrate molecule by a non-hydrolytic process. Notable examples include decarboxylases, aldolases, and syntheses, such as citrate synthase. Isomeroses interconvert positional, geometric, or optical isomeric structures by intramolecular rearrangements. Notable example includes mutases, such as methylmalonyl coenzyme mutase. Ligases join to separate molecules by creating a new chemical bond at the expense of a nucleoside triphosphate molecule. Notable example is the synthetase group of enzymes. Synthesis process by synthetase enzymes is coupled with hydrolysis of a phosphoanhydride bond of ATP or some other energy-rich compound, DNA ligase, fatty acyl coenzyme synthetase, glutamine synthetase, amino acyl tRNA synthetase are notable examples. Some authors refer to another new class of enzymes which catalyzes translocation of ions and molecules across membranes by, or without, using the energy of hydrolysis of ATP. This is the translocase class of enzymes and includes specific enzymes for transferring inorganic ions, hydrogen ions, amino acids, carbohydrates, cations, etc. Notable example is ATP, ADP translocase. Isoenzymes or isozymes are closely related enzymes that catalyze the same reaction but differ in their molecular structure, electrophoretic mobility, kinetic properties, sensitivity to inhibitors, response to allosteric modulators, tissue origin, and other properties. In short, isoenzymes have different structures but identical function. Allozymes are variant forms of enzymes encoded by different allos of the same gene. Through allozyme analysis, it is possible to detect functional genetic variation without resorting to expensive molecular and genetic testing, and therefore allozymes are considered as important markers in population genetics and evolutionary studies. Allozymes can be distinguished by their electrophoretic mobility where they appear as discrete bands. Allozymes have been mentioned here so that they aren't confused with isozymes. Isozymes have more importance in medical science and diagnostics. After acquainting ourselves with the foundational concepts and terminology, we now arrive at the juncture where we inquire, what is the mechanism by which enzymes operate? How do enzymes work? How do enzymes perform such extraordinary catalysis? How do enzymes enable conversion of thousands of molecules within fraction of a second? Firstly, it's very important to understand that chemical reactions occur because atoms and molecules interact with each other, leading to the formation of new substances. This happens due to the rearrangement of chemical bonds, driven by factors such as the need to achieve lower energy states, or reach a more stable configuration. Whenever atoms and molecules interact, they will invariably seek to achieve lower energy states or increased stability. Hence, thermodynamics favors the formation of product molecules whenever reactant atoms or molecules interact. However, even though nature favors the formation of products, certain conditions need to be fulfilled. The first condition is that bonds in the reactant species need to be broken. There's also the need for alignment of reacting groups, formation of transient unstable charges, and other transformations. This invariably requires input of extra energy. This extra energy is termed as the activation energy. Secondly, meaningful reaction can occur only if sufficient number of reacting atoms or molecules can collide with enough force and in the precise orientation required for existing bonds to break. If only bonds get broken in sufficient numbers, atoms or molecules can rearrange themselves to form new bonds, resulting in the creation of different substances. Therefore, in most cases, an energy barrier exists, which the reacting atoms and molecules need to overcome. In other words, the atoms or molecules of the reactants need to gain sufficient energy, so that existing bonds can be broken. Furthermore, sufficient number of atoms and molecules would need to collide with each other in precise orientation. All this could be achieved by raising the temperature, or pressure, or the concentration. However, in the biological system, this is an impractical solution. The other way to do this is to find an alternative reaction pathway where less activation energy would be required, and also sufficient number of molecules could be made to collide. This is exactly what biocatalysts do. By some means, enzymes are able to provide an alternative reaction pathway for the reacting molecules. In the alternative pathway, less activation energy is required. Obviously, it now becomes very easy for existing bonds to break down and new bonds, which favor more stability and low energy states, to start forming. In other words, new product molecules can form. Enzymes also enable more molecules to collide, thereby effectively increasing the probability of bond breakage and formation. There's a saying that, in science, answers sometimes lead to more questions. The question that arises now is, how exactly do enzymes create a new reaction pathway where less activation energy is required? And how do enzymes ensure that more atoms and molecules, rather than less, collide with each other with sufficient force? This is where the transition state comes into picture. Transition state of a reaction is the high-energy momentary intermediate formed during the conversion of reactants to products. It occupies the top of the energy hill and can flow in either direction. The reason that activation energy is often high is because of this unstable transition state that mandatorily gets created during chemical reactions. Much energy is required to carry reactant atoms and molecules till the transition stage, only to see them reverting back to reactant stage. This can be pretty frustrating. But what if that transition state can be stabilized and nudged to move only in the direction of product formation? The entire reaction can then be made to proceed easily in the forward direction. Well, enzymes do just that. They stabilize the transition state through a number of mechanisms, and moreover, they also employ those same mechanisms to bring more reactant atoms and molecules to collide. This stabilization can occur through various mechanisms, including electrostatic interactions, hydrogen bonding, and induced fit conformational changes in the enzyme-substrate complex. The end result is that more reactant atoms and molecules reach the transition state, and those transition state species run fast forward towards converting fully into stable product molecules. The goal is thereby achieved. The induced fit model, as mentioned in the previous section, proposes that the enzyme undergoes conformational changes upon substrate binding, resulting in a more optimal fit between the enzyme and substrate. This conformational change stabilizes the transition state and lowers the activation energy required for the reaction to proceed. But it's very important to understand that apart from stabilization of the transition state, the other mechanisms of lowering activation energy during catalysis involves precise substrate orientation, direct participation of the enzyme in the reaction, conformational changes upon substrate binding, as described in Kochelin's induced fit model, and the involvement of cofactors and coenzymes. These other mechanisms employ acid-base catalysis, covalent catalysis, and metal-ion catalysis. In acid-base catalysis, enzymes donate or accept protons to facilitate the reaction. Covalent catalysis involves the formation of a transient covalent bond between the enzyme and the substrate. Metal-ion catalysis utilizes metal ions bound to the enzyme to facilitate the formation of reactive intermediates or stabilized charge transition states. All these mechanisms result in less activation energy requirement. All activity occurs at a special site in the enzyme molecule known as active site. Enzymes provide a specific and protected environment within the active site where a given reaction can occur more rapidly because of lowering of activation energy by various mechanisms as described in the foregoing section. Furthermore, the active site enhances the probability of productive atomic or molecular interactions. Substrate molecules get bound to the active site and forms enzyme-substrate complex. In closing, enzymes stand as remarkable catalysts, driving essential biochemical reactions in living organisms. Their precision, versatility, and efficiency underscore their paramount importance in sustaining life. As we continue to unravel the intricate mechanisms governing enzyme function, we gain invaluable insights into both biological processes and potential applications in various fields, from pharmaceuticals to diagnostics.
Thanks for watching this video. Our next tutorial in the Medical Biochemistry Power Capsule series is titled Enzymology, Basic Principles of Activity and Kinetics. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and share with your friends. Until next time, do keep exploring.